For many years, extracellular matrix, or ECM, was dismissed as a relatively inert ground substance whose sole function is packing. However, it has now become clear that the ECM is much more complex than was once supposed, and that interaction with the surrounding matrix is one of the major controls of cell behavior. In plants, ECM appears mainly as cell walls, but in animal tissues it appears in two main forms: basement membranes and stromal matrix.
There are over 100 known extracellular matrix components. The ECM is mainly composed of an intricate interlocking mesh of fibrillary and non-fibrillary collagen, elastic fibers, and glycosaminoglycan containing non-collagenous glycoproteins that forms a highly cross-linked gel. Although collagens are collectively the most abundant component of the ECM, the differential expression of individual interstitial ECM components underpins the specific functions of many organs and tissues. The extracellular matrix serves as a structural element in tissues and also influences their development and physiology.
Cells are not just suspended in or resting on the ECM. Cells embedded in the ECM make contact with it through specialized matrix receptor molecules. These allow the cells to adhere to the matrix and may also signal the presence of particular matrix components to the internal machinery of the cell, allowing the matrix to control cell behavior. And the most important of the receptors are integrins.
Integrins are involved in adhesion of cells to the ECM and to each other. Until integrins are bound to the ECM, they diffuse about freely on the plasma membrane. Once bound to an ECM, they cluster and start to form a protein plaque at the cytoplasmic base of the integrin. The complex of integrins in the cytoskeleton is called a focal adhesion.
An extracellular binding site allows for cell adhesion because of the alteration of an integrin through intercellular signaling.
Two epithelial layers along the basement membrane make up the human breast. This helps to create the branching ductal network. The luminal epithelial cells are in an inner layer, and the myoepithelial cells are the outer layer. The basement membrane then surrounds these layers.
Mammary glands go through multiple morphogenic changes throughout puberty, pregnancy, lactation, and post-lactational evolution. At puberty, the mammary ducts extend outward into the surrounding fatty tissue of the breast. This also occurs during pregnancy.
An ECM acts as a basement membrane and surrounds the epithelial cells, which are formed into a somewhat spherical cyst that secretes milk. If the cells are not in the presence of an ECM, so if the cells were placed in a culture dish, they will cease to produce milk, even if there are appropriate hormones, and will eventually die from apoptosis. If the cells are in an artificial ECM that contains laminin, the cells still form a cyst and look very similar to a real mammary gland. They will survive and produce milk normally.
It is now known from studies that the cells bind laminin via integrins, and that this causes a domino effect that causes milk production. When milk production is no longer needed, the alveoli are destroyed and will be formed during the next pregnancy. Matrix metal proteinases are a group of enzymes that break down the matrix, which is then followed by the alveoli destruction, which suggests that milk production is largely affected by the ECM.
This study was done to show how cell matrix interactions control outgrowth of mammary epithelium during puberty and pregnancy. They hypothesized that similar to embryonic aortic smooth muscle development, Fibrin 2 and VICAN expression alters the cell matrix interaction to allow mammary ductal outgrowth and development during puberty and to enable epithelial budding during pregnancy.
The following images are tissue sections of pregnant mother's mammary glands. P1 displays 24 hours after segmentation. P2 is 48 hours after. P3 is 72 hours after. P4.5 is 4.5 days after. And P12.5 is 12.5 days after. The images on the right-hand side are a magnification of the asterisk site on the left side. Fibrin 2 starts to increase after 48 hours after conception. It peaks at 72 hours and begins to decrease after that. So now we're going to show you some videos comparing an inactive breast histology slide with an active breast histology slide.
This is human breast tissue. A good general rule of thumb is that when you look at a breast tissue from the correct perspective, which is the way we're looking at it right now, you know that all of these little blue areas are lobules. There's a lobule, there's a lobule, there's a lobule, there's a lobule. You know that all of these areas which are basically pink, which are connective tissue, is called stroma. There's stroma, there's stroma, there's stroma, there's stroma. The rule of thumb is that if the stroma is about the same or even more abundant than the lobules, this is probably a woman who is not producing much milk. This could be somebody that is postmenopausal. And basically, the more postmenopausal you get, the higher the ratio between the stroma and the glands or the globules. This is somebody who is not producing milk. It could be somebody postmenopausal. Also notice that when you look within the lobules, you do not see any secretions either. These are ducts of the breast. Here's a duct within the lobule. Here's a duct between lobules. Here's an artery. Here's some fat. There's always a lot of fat in the breast. And at the next lower phase, lower than lobule, you have these individual structures here and here. And these are the actual acini. Sometimes they're called alveoli, and each acinus is separated by a small amount of connective tissue from other acini as well. Otherwise, this whole thing is a lobule. And sometimes, if you see a structure like this, which is strictly lined by perhaps a simple cuboidal type of epithelium, you know that this is a duct. And if you see an area like this, this is an acinus. And the acinus makes the milk in the ducts, whether they're intralobular or extralobular, they just transport the milk. The ducts, the acini have basically two kinds of cells. There are the actual epithelial cells, which are making the milk product, and then there will be little spindly cells, like here, maybe here, possibly here, right at the periphery of the acinar groups, and these are the myoepithelial cells. The myoepithelial cells contract, and they assist the epithelial cells to express the milk product through these ducts, first intralobular, and then ultimately into larger ducts, which are between the lobules, like perhaps here. We'll find one quickly, I hope. Like here. And then ultimately into larger ducts, and ultimately through the lactiferous ducts through the nipple. Well, this is shotgun histology, and is there anything here you can't identify? You can identify lobules. You can identify small acini. You can identify ducts. You can identify bigger ducts. You can identify connective tissue called stroma, chiefly fibroblasts and collagen. You could identify fat. You could identify large arteries. You could identify veins or perhaps lymphatics. And I really don't think there is anything on here that I can click on a random field that you can't identify. And for that reason, that is why I'm going to say thank you very much.
So this is the slide of the active breast tissue.
This is some more human breast tissue. And in deference or in contrast to the last breast tissue that we saw, which was inactive, which means it is not pregnant or lactating. We have breast tissue here in which the lobules look like they are considerably more in cross-sectional areas than the surrounding fat or connective tissue. So this is what we call an active breast. This could be perhaps a pregnant woman or an early lactating woman, but usually in lactation, these little acini are much bigger. So I suspect this is only a pregnancy. And in the active breast lobules, you can see that the acini are filled with proteinaceous material. This is called milk. And if you took a drop of milk and stained it with hematoxylin and eosin, the proteins in it might look very much like this. Here's a little bit of very loose connective tissue surrounding these lobules. Here's denser connective tissue and fat between the lobules. Otherwise, most of these acini within the lobule are now actively producing milk. And here we see that virtually all these acini are distended with milk product. We can see a duct inside of one of these lobules, as well as a duct at the periphery of the lobule. Remember, the duct is just strictly transporting the material, whereas all of these acini are actually making the juice. Here's a little blood vessel between the lobule. Here's another one. Here's some fat. Here's some fibroblasts. And you can quickly see that I think we've pretty much nailed down this whole breast. Let's find a large duct, perhaps. Probably here are a few larger ones, but let's see if we can find something larger than that. Yeah, I think you could say this is a pretty large size duct, and look how it is filled with milk as well. So here's the lobule. Here are the distended alveoli. The alveoli still have the myoepithelial cells at the periphery of them. Sometimes alveoli are also called acini. Here is loose connective tissue under the influence of estrogen supporting these delicate lobules. And then here is denser connective tissue and some fat and more fat and a lot of fat composing the stroma. And because the stroma here looks like it's probably less than the lobules, you know this is an active breast. And I thank you very much.
The take-home message from this presentation is that the production of milk is a complex process that relies on multiple things working together, but without the extracellular matrix binding to the cells to influence cell behavior and prevent apoptosis, milk would not be produced at all.